About 1 century ago, Hans Berger recorded the first EEG recording in humans. He was a German psychiatrist, and in the picture you see his lab in Jena, the equipment for the recording of EEG in humans from one channel. The importance of Hans Berger is really great in the field of quantitative and EEG, because he founded the terms and the main concept of resting state EEG activity in humans. He was the first to name electroencephalography, and was the first to name the main rings in the EEG activity during a resting state, eyes closed and eyes open. And he called alpha and beta waves the main rings in this condition. The first recording was in 1924, but he preferred to collect more data before the first publication of a human EEG in 1929. He proposed that EEG activity is a biomarker of mental states in brain diseases, with dysfunctions in vigilance, cognition, and was also one of the first researchers to show the epileptiform activity in humans with this technique. The resting state condition implies that in the instructions, the experimenter asks for a waiting psychophysiological relaxation in the participant. And he was asked to mind wandering, to be relaxed, not to perform voluntary actions during the recording. And the typical condition was eyes closed for a few minutes, and then the instruction to open the eyes for another while. And you see in the picture the relaxed posture of the participant, and the cap with the electrodes, the amplification, and typically the experimenter control the EEG activity, the ongoing EEG activity during the experiment, and also the general behavior of the participant. When you record EEG activity in the resting state, eyes closed and eyes open condition, you are probing the brain circuits regulating the vigilance, the consciousness level. And the vigilance consciousness level is one important dimension of a consciousness. In this graph, you see that you have in the vertical axis the level of consciousness and vigilance, and in a horizontal axis the content of consciousness or vividness of the contents of consciousness. The contents of consciousness are the images, the mental images that we have, the thoughts, the thinking process to elaborate memories and so on. And you see that in the horizontal axis, you can see in the axis the condition in which you are with a sort of blank in the mind, or more images in the mind, or an intense imagery activity. In the vertical axis, you have the level of vigilance. And you see that from the lower part of the axis, you have a very low level of vigilance, the deep sleep, for example. And increasing the level of vigilance, the level of or consciousness, you see that you have a light sleep, the condition of quiet vigilance, typically of resting state condition, and then more higher, more and more higher vigilance conditions to alertness and intense concentration. And so you can define the general level of consciousness and the vividness of contents of consciousness, and put the participant in a place in this graph during your EEG recording. Here you see the two main electrode montages to record EEG. EEG activity derives from a differential measurement of electric field, typically on the scalp. And you see in the left the example of a bipolar EEG recording in which you have a positive electrode over the vertex in this example, and the negative electrode in the posterior scalp region. So the two electrodes, positive and negative, are closing a loop you see here, and then they are amplified and recorded typically in a computer. And so what you measure is one channel from the two electrodes with the EEG activity. In the lower part left of the figure, you see that you have 4 scalp electrodes, and you can set a bipolar montage in which you have that 2 electrodes derive one bipolar channel. And in this case, you see that if the source of EEG activity is in the center of these circles, you can record with bipolar electrodes no EEG activity over the source, but you can record up and down with opposite polarity. You see the vertical dot line, and you see that you have an upper peak in the upper bipolar channel and a lower peak in the lower bipolar channel. And this helps you, this reversal of polarity, to identify source of EEG activity in the middle. The other typical electrode montage, which is used mostly for research application with the mapping of EEG activity from a higher number of electrodes, is the unipolar electrode montage. And you see that the positive electrode is over on the scalp, and the negative electrode is placed in the hair lobe, in this case, so in a place far from the cerebral cortex. And the typical placement of the reference electrode is on one part of the body, closing the contact with the positive electrode, but far from the sources of EEG activity in the brain. So ideally, one can assume that there are no sources near the reference electrode, the negative electrode. And you see that the loop is closed, the one channel of EEG activity is amplified and recorded. And you see that this kind of unipolar EEG electrode montage allow you to record waves in the contacts, and they typically have a similar amplitude and similar phase at all scalp electrodes due to the volume conduction of the neural currents across the head. And so you see that you have a more continuous amplitude and a similar phase of the waves with the unipolar montage. So you see in this picture the different spatial scales at which you can record electrophysiological activity with a positive and a negative electrodes. Our lecture today is focused on EEG activity, which is recorded typically with the scalp electrodes, but in some cases for clinical applications, electrodes can be implanted in the cerebral cortex or even in subcortical structures. For example, for clinical applications in epilepsy, in the case of pharmacological ineffective therapies in the epileptic patients, requiring the removal of the brain sources of epileptiform activity. In this case, the electrodes can be implanted over the surface of the cerebral cortex with the subdural electrodes, or even with the stick electrodes in the layers of cerebral cortex, or even in the white matter under the cerebral cortex, with electrodes implanted also in basal ganglia, nuclei, or other subcortical structures. And when you put an electrode over the scalp or in the surface of the cerebral cortex, you obtain the typical waves of EEG activity represented in the figure. And you see that the regions generating EEG activity at one exploring electrode is really relatively large. And you see that you are talking about millions and millions of neurons active simultaneously to produce currents, ionic currents, so big to be able to change electrical field at the scalp level. But electrophysiological recordings can be obtained also on a smaller scale, spatial scale, a mesoscopic scale, and to capture the electrophysiological activity of populations, in this case, population of neurons in the brain. And in this case, you use the term of local field potentials. In the local field potentials, so you have the frequencies of the oscillations that you can find in the EEG, but even higher frequencies of, for example, 1 Hertz or 200 Hertz or 300 Hertz in the recordings in relation to the ability of these electrodes within the cerebral cortex or brain structures to capture the electrical activity of small populations of neurons. And of course, you can also have electrophysiological recording at microscopic scale, for example, in the extracellular space of a single neuron. So you are able to capture action potential spikes or even changes in the postsynaptic potentials. So one point of research was to understand the extent to which the EEG activity is related to local field potentials or spikes or postsynaptic potentials. And the research showed that the EEG activity is mainly related to postsynaptic potentials from a very large number of cortical pyramidal neurons. In this graph, you see the temporal and spatial features of EEG activity. In the vertical axis, you have the spatial resolution of several brain research techniques. And in the horizontal axis, you have the temporal resolution for the recording of brain activity. And so you see that the techniques with the lower spatial resolution are the techniques allowing the recording of action potentials or postsynaptic potential. They are microelectrode electrophysiological recordings, ME, and so you can have a spatial resolution of a fraction of a millimeter. And then you see that you can have with the resolution of 0.1 millimeter with electrode array of put in the cerebral cortex, and so higher with local field potential. And for example, 1 millimeter, if you put an electrode on the surface of the cerebral cortex, and when you go to electroencephalographic activity recorded from scalp electrodes, you have a higher indetermination of the sources. And you see that you have here 1 and more centimeters of spatial resolution with EEG activity. The magnetoencephalographic activity is able to capture the brain neural activity with a higher spatial resolution of some millimeters. So EEG, you see, has a low spatial resolution to localize the brain activity. So this is the weak point of EEG activity. The strong point of EEG activity is the high temporal resolution. So you can record EEG activity every millisecond if you want to, with 1,000 hertz in the sampling of EEG activity. And this allows you to measure the oscillations of brain neural activity at high frequencies. When you compared EEG with functional magnetic resonance imaging, you see that the functional magnetic resonance imaging has 1 millimeter of spatial resolution of brain activity, so a high spatial resolution, but a low temporal resolution. You can have one map of functional magnetic resonance imaging every second, which is a long period in relation to the dynamics of brain activity. And so we can come back to the first experiments of Hans Berger. And you see here the typical layout of Hans Berger's experiments. He was not able to store the human EEG activity at the time, and so he has to take pictures on the EEG activity on the oscilloscope. And you see that in the left that he used a very simple one bipolar channel with the positive electrode in the frontal scalp region and the negative electrode in the posterior scalp region. You see that the loop of the electrodes, the amplification, and the visualization of EEG activity in one oscilloscope. And this is one of the typical condition of EEG recording. So Hans Berger was the first neuroscientist to use the resting state condition for the recording of brain activity. And he introduced the condition of eyes closed and eyes open. And in the picture, the green frame is used to emphasize the period of eyes closed condition, and the blue frame for the eyes open condition. So when you see the skull, the time scale, 1 second, and so if you count the peaks in the EEG oscillations, you see that you have the 10 Hertz condition. And so you see the ample EEG activity at about 10 Hertz, and he termed these oscillations alpha rhythms. When the subject opens the eyes, you see that there is more cortical activity related to visual information processing. So there is an increase of the vigilance, and the alpha rhythms disappear. And so you don't see the ample oscillation at 10 Hertz. And Hans Berger described this higher frequencies appearing, and he called them beta rhythms or beta waves. And here you see that if you count the peaks, you see that is 20 or more cycles per second. When he asked to close the eye again, and alpha oscillations appears again. And then he was the first to demonstrate that this is really the spontaneous oscillation of brain activity in the resting state condition, because he could demonstrate that these oscillations were not due to electromyographic activity or respiration, and they were not related to beats of the vessels and heart rate. And this was the layout of the first publication in 1929 in a journal of psychiatry published in Germany. When he published the first results of the human EEG activity, there was a very strong discussion if this finding was grounded in the brain activity or was due to artifacts induced by the equipment. So the Nobel Prize Adrian, who received this award in 1932, so 3 years after the discovery of human EEG by Hans Berger, and the first publication is 1929, he decided after the Nobel Prize to be engaged in this controversy at the time if the human EEG could be recorded following the Hans Berger's procedure. So Adrian wanted to replicate the Hans Berger's experiment. So together with an engineer, Matthews, was able to set up an EEG recording from humans following the Hans Berger's procedure, eyes closed and eyes open. And he was able to confirm that there is a spontaneous, dominant alpha rhythm in the resting state, eyes closed condition. So you see the results published by Adrian, and you see that in the eyes closed condition, he was able to record an ample alpha rhythm. And when the participant opened the eyes, the alpha rhythm disappeared. When he asked again to close the eye, the alpha rhythm appears again. And he was also to demonstrate that the other kind of increase in the vigilance and in brain activity were related to the reduction of alpha rhythms. In this case, asking the patient to be engaged in a visual spatial task. And this was the another important paper by Jasper reporting the results of the first human EEG recording in United States of America. And he published this finding in 1935. The publication by Adrian was 1934. The particular finding by Jasper in the publication of 1935 was that he used not only eyes closed condition and was able to replicate the dominant alpha rhythm. In this case, you see that he recorded and published the results also of electrocardiographic activity, and was able to demonstrate there is no relation between alpha rhythms and heartbeat. And so he could demonstrate again a dominant alpha rhythm in the resting state condition, but he introduced a nice experiment with visual stimulation with the flashes. And so he put flashes and could record the EEG changes related with visual flashes. But when the visual stimulation was switched off and the subject was in the eyes closed without any visual stimulation, immediately after the switch off of visual stimulation, he showed a reduction of alpha rhythms and the appearance of beta oscillations. So in other words, he could demonstrate that the reduction of alpha rhythms related to cortical activation and increase of vigilance could be induced immediately after the visual stimulation, when this in the mind of this participant there was again some after effects of visual stimulation. So he was the first to say that even if you are in the eyes closed condition, in the resting state, immediately after visual stimulation, when you again have some after visual effect, and so there is a after effect visual information processing in the brain, even if the visual stimulation was switched off, you have the reduction of alpha rhythms. So in some way, this was one of the first demonstrations that the mental activity is related to alpha rhythms, the reduction of alpha rhythms. And so he could demonstrate that this is a short period after the visual stimulation, and then after a few seconds, you see again the alpha rhythms appeared again. So in some cases, you can probe the mental activity in the participant as measured by the amplitude of alpha rhythms. After this exciting period for the EEG science, Hans Berger, Adrian, Jasper, many other researchers involved in the demonstration of the possibility of EEG techniques, there was another important chapter in the field of neurophysiology, human neurophysiology, the use of EEG techniques to explore the transition of vigilance from quiet wakefulness to drowsiness, to light sleep and deep sleep. In other words, EEG techniques were used to investigate the brain activity related to transition to sleep and sleep. And This was an important field also for clinical applications on sleep disorders over the decades. And today, EEG is the most important technique to investigate sleep disorders in humans. Here we have a milestone research from the group of Hori. You see in this case that we have an EEG recording with a bipolar electrode montage. And in the picture, you see that if you go down in the graph, you see decreasing brain activation, brain arousal, decreasing of vigilance level. So from one channel, you record EEG activity during this transition from wakefulness to light sleep. And from the same channel, you can see the EEG oscillations, the frequencies and the amplitude of these oscillations changing from the same scalp electrode in the transition from wakefulness to light sleep. And this is the power of EEG activity, that you can, analyzing the frequencies and the amplitude of EEG activity, you can derive the mental state of the human in the transition from wakefulness to sleep. And you see that if you see the skull, the time scale, 1 second, and if you count the peaks in 1 second, you see that there is an alpha frequency dominant in the quiet wakefulness, resting state, eyes closed. But you see that during the recording, the epoch of EEG recording show some fluttering of EEG activity, low amplitude, and some disappearance of the dominant alpha oscillations. And during even more transition to drowsiness, you see that in this epoch, for example, of 3 or 4 seconds, you see that the period of fluttering are longer. And this reflects the transition from quiet wakefulness to drowsiness and light sleep. And in some cases, during this transition, you see a really flat EEG activity, which reflects the beginning of a very light sleep. And then during this transition to light sleep, you see that you have a slowing of the frequency of EEG activity from the same scalp electrode. And this is a frequency lower than alpha, is a theta rhythm lower than 7 Hertz that we called ripples of sleep. And when the sleep becomes deeper, a bit deeper during this transition, you see the appearance of very ample waves that we call key complexes. These are really big peaks showing the periods of no mental activity, of a sort of blank of mental activity related to sleep. And finally, there is another very clear EEG wave of the sleep, which is represented in this spindle EEG activity. And these are called spindles of sleep EEG. And so key complexes, vertex positive waves are, and spindles are very clear waves of non-rapid eye movement sleep. And so you see that the power of EEG technique, you can put just 2 electrodes, positive, negative. You can obtain one wave in this bipolar EEG montage from the same place, and during the time in relation to the changes of the vigilance level from wakefulness to sleep, you can observe from the same channel, from the same EEG trace, the changes in the frequency and the amplitude, reflecting the vigilance and consciousness level. These experiments of Horry are a reference in the literature, in the history of EEG science, because he correlated these changes in the EEG frequency and amplitude with the threshold to awake the participant. So Horry and his co-workers observed the EEG activity on the screen during the transition from wakefulness to sleep. And when they see, for example, fluttering, they ask for a question to the participant, and in some cases do not obtain an answer because the subject was in a sort of light sleep. But if they ask for a question or ask for a reaction during the key complexes or spindles, they obtain less reactions from the subject. So they could demonstrate that there was an increase of inhibition of the brain to react in line with these EEG features. You see that until now, the EEG analysis was based on the observation of the EEG waves and counting the peaks to define the frequencies, to analyze the amplitude, and to derive the mental state of the participant in terms of vigilance level from wakefulness to sleep. But an important step forward in the field was to obtain quantitative measures of EEG activity. And so you see that we have the bipolar electrode montage. We derive one bipolar EEG activity over the time, 1 second, for example. We can count the peaks and to say that there is a dominant alpha oscillation at 9 Hertz. But the step forward was to apply a mathematical analysis of these oscillations. And this was done with the fast Fourier transform, a mathematical approach enabling to derive the power of the oscillations in the EEG activity. So you can imagine that this technique is able to decompose the true EEG activity in elementary oscillations. 1 Hz, 2 Hz, 3 Hz, 4 Hz, and so on. And to measure the amplitude from the upper peak and the lower peak, to square this amplitude to obtain the power density. And so, for example, the fast Fourier transform from the elementary 9 Hz oscillations within the true EEG activity is able to capture these oscillations and to square and to put the result in the so-called power density spectrum of EEG activity, in which you have in the horizontal axis the frequencies. In this case, in this simple example, we have just 9 Hz, and in the vertical axis, the power density of EEG activity. And with this technique, you are able to identify immediately which is the frequency showing the highest power density in the alpha band from 8 to 12 Hz, and to identify the individual alpha frequency peak in the EEG power density. And so you can have an individual analysis of alpha frequency in a given participant. And this is another example. For example, you can imagine that you have a resting state condition. You record this in eyes closed condition, and you obtain this through EEG. This is an example with an EEG activity based only on 3 elementary oscillations with a given amplitude and a given phase of the oscillation. And you see that the fast Fourier transform can decompose this EEG activity recorded from one channel and to derive the 3 oscillatory activity, the 3 elementary sinusoid of EEG activity. In the example, one is 1.5 Hz, the other 8 Hz, and the other 20 Hz. 1.5 Hz is in the so-called delta band of EEG activity, lower than 4 Hz. This oscillation at 8 Hz is within the alpha band from 8 to 12 Hz. And this oscillation of EEG activity at 20 Hz is in the so-called beta band from 13 to 30 Hz. And so the fast Fourier transform is able to decompose the original EEG in oscillatory oscillations to compute the amplitude peak to peak, to square, to obtain the power, and to put the results in the power density spectrum of EEG activity. Again, the vertical axis, the power density, in the horizontal, the frequencies, and you represented the results. In this case, showing that in this EEG recording in this period, the dominant power density is in alpha, and then delta and beta. Another evolution of the quantitative analysis of ongoing EEG activity was focused on the interdependence of EEG activity in different scalp regions. So the idea is that if you have this interdependence from a statistical point of view, you have a sort of functional connectivity of the generators of this EEG activity. You see in the this graph that we have 2 bipolar channels. One bipolar channel over the frontal scalp region, producing EEG activity. Another channel is on the parietal areas. And so you see in the bipolar channel and an EEG activity from these parietal bipolar electrodes. And so you have in the same recording two EEG activities, one from the frontal bipolar channel, the other from the parietal bipolar channel. And the analysis of these two waveforms is called coherence analysis, spectral coherence analysis. There is a decomposition in the frequencies. In the example, 1.5 Hz in delta, 8 Hz in alpha, and 20 Hz in beta. From the frontal bipolar channel, the parietal bipolar channel, and the spectral coherence analysis show if you have a correlation of the phases of these two oscillations in each frequency. If you have the same phase, so the simultaneous, for example, course or a strictly correlated course of the two waveforms, you have the maximum coherence that in this normalized measure is 1. If the phases of the two oscillations are not related during the recording, so they are continuously changing, you have 0, which means no functional collectivity between the sources of the two EEG waveforms. And so you can investigate if there are interdependencies between two waveforms in 1 or 2 frequencies or more frequencies. In the case of this example, we set the maximum interdependence in the 3 frequencies between EEG recording from frontal and parietal bipolar channels. And so on the basis of this coherence spectrum of EEG activity in two electrodes, you can map the functional connectivity between cortical regions during the EEG recording. So you can investigate the networks in the brain. So another step of the EEG research was to localize the sources of scalp recorded EEG activity, because when you record the EEG, you are measuring the electrical field under the positive electrode in relation to the negative electrode, instant by instant, to generate the EEG activity over time. But the point is, where is the source of the scalp recorded EEG activity? So the electric field on the scalp is due to the activity of the neurons in some places in the cerebral cortex, maybe in structures under the cerebral cortex. So the point is, where is the source? Where are the sources of EEG activity? Today, we know that the sources of EEG activity recorded at the scalp level are in the dendrites of the cortical pyramidal neurons, because the main dendrites of the cortical pyramidal neurons have this particular orientation spatially. They are aligned as a palisade in the cerebral cortex, and they have this perpendicular orientation in relation to the cortical surface. So this is a very convenient spatial orientation for the spatial summation of the postsynaptic potentials under the terminals of the axons of these neurons. So you see here that you have several terminals, the currents, ionic currents from the terminals of the axons and the dendrites. The current, the neurotransmission, so you have binding of neurotransmitters at the level of dendrites, and you have the opening of the channels at the level of the dendrites, and you have the currents of ions from the extracellular space into the dendrites, and also some ionic currents from the dendrites to extracellular space. So you have the loop of these ions and this the extracellular currents of the ions related to postsynaptic potentials are the main source of scalp recorded EEG at the level of these the main dendrites of the cortical pyramidal neurons. Other neurons, for example, interneurons, do not have this spatial orientation of the dendrites. So the currents from the dendrites of the interneurons, or the currents of ions from extracellular space into the dendrites, these loops of ionic currents cancel each other. So you can have very active interneurons in the cerebral cortex, but they do not have the convenient spatial orientation to generate extracellular ionic currents able to summate and to change the electric field at the scalp level. And this is an example of the spatial summation of postsynaptic potentials and the related ionic currents. You see that in this example, you have a simultaneous neurotransmission from terminals of axons and so action potential 1, 2, and 3, and you see that there are some related ionic currents, and this spatial summation of postsynaptic potential, ionic currents within, but also in the extracellular space, you have passive electrotonic ionic currents, and these extracellular electrotonic currents produce the generation of scalp recorded EEG. And this is another example. You see here the case of cortical pyramidal neurons with the main dendrites, and you see that you have the case of 3 synaptic contacts in these three pyramidal neurons. You see that when the contacts are in the proximal part of the dendrites, you can generate here excitatory postsynaptic potentials with cations entering into the dendrites, and you can imagine the extracellular electrotonic ionic currents here in the red arrows, and there is a summation of these extracellular ionic currents, and these changes and summation of these ionic currents is related to changes in the electric field at the scalp level. And so here the positive electrode, the negative is not shown, is sensitive to this summation of extracellular ionic currents related to postsynaptic potentials at the same time. And you see the deflection of EEG activity. In the example, you see the lower deflection related to this neurotransmission at the proximal levels of the dendrites. When you see that the other case in which the synaptic contacts are in the distal parts of the dendrites and the more close to the electrode, again you see that the cations get into the dendrites during the excitatory neurotransmission, and so you see here the arrows are the electrotonic ionic currents in the extracellular space around the main dendrites of the cortical pyramidal neurons, and so you have a change in the electric field with the upper deflection of EEG activity related to this different place of the synaptic contacts. So if you compare the two cases, you see that the deflections, the lower deflection or the upper deflection of EEG are the summation of these synaptic events at the level of main dendrites of cortical pyramidal neurons, with a mix of cases that of course make complex the frequencies and the amplitude of EEG waves. In this picture is illustrated another important feature of the sources of scalp recorded EEG. So you see in the example, the scalp electrode, the positive electrode, the negative electrode is not shown, that measure the electric field under the electrodes. And you see here the scalp, the skull, the meninges, and the cerebral cortex with the cortical pyramidal neurons. Again, you see the main dendrites at palisade perpendicular to the cortical surface. In the middle of the picture, you see the case of several synaptic contacts with the main dendrites of cortical pyramidal neurons. And you see that the neurons are numbered 1, 2, 3, 4, 5, 6, and in the right side of the picture you see the postsynaptic potentials for the neuron 1, the neuron 2, the neuron 3. And so in some cases you see that you have some postsynaptic deflections in the potential at the membrane level of these dendrites. And in some cases, you have this desynchronization related to excitatory postsynaptic potential, the upper deflections. But it's clear that the timing of these postsynaptic potentials, the time of these deflections, are not at the same time, are not synchronized from a temporal point of view. So that when you have the summation, the spatial summation of these postsynaptic potentials, you don't have very ample potentials. You don't have very ample voltages. And so you are really flat potential related to the spatial summation. So we say that in this case, you see the activity of the cortical pyramidal neurons, but non-synchronized from a temporal point of view. So when you see ample EEG at a given frequency, you are observing the temporal synchronization of the cortical pyramidal neurons at that frequency. If you don't have a synchronization, you don't have an ample EEG activity. In the lower case, you see that the desynchronization of the cortical pyramidal neurons during the excitatory postsynaptic potential, you see the upper deflection in all neurons at the same time. So they receive the neurotransmission from other neurons at the same time of the main dendrites. And so you see the excitatory postsynaptic potential and the synchronization at the same time. And the spatial summation of the postsynaptic potential at the same time generates these ample changes of scalp recorded EEG activity. And so you see the peaks, upper peaks or lower peaks in the oscillation of EEG activity in the relation to synchronized activity of the cortical pyramidal neurons. So what we are exploring with the EEG techniques is the synchronization of cortical pyramidal neurons at the different frequencies. And at different frequencies reflect the general cortical arousal, the general cortical activation, for example, during quiet wakefulness or alertness or drowsiness or light sleep or deep sleep, non-REM sleep, or in the rapid eye movement sleep with vivid dreaming. And so we have another demonstration that when we talk about the EEG waves with some frequencies, a given amplitude, we are talking about the synchronization of postsynaptic potentials at the level of cortical neurons. So here we have a very nice case, in which we have in dogs in the visual cortex, the simultaneous recording of single neural activity. And you see these dots. Each dot is an action potential. You see the typical raster in which you have different electrodes, in array of electrodes, several contacts are able to record extracellular action potential. So you see this population of cortical pyramidal neurons. And you see that when you have this continuous activity, but not synchronized in different neurons, so you don't see a rhythmic action potential activities in these different neurons, you see that you have a flat scalp recorded EEG. But when you see some rhythmic synchronized activity from action potentials in this group of neurons, you see that you have a very nice relation between synchronized excitatory activity with the action potential at the same time in different neurons. You have a nice correspondence with the peaks of scalp recorded EEG. And another important aspect of this picture is that you see that this rhythmic activity derive from short periods of intense neural activity with synchronized action potentials in this neural population. And this is nicely related to the lower peak of EEG. And when there is a silent without action potentials in this neural population, each line is a neuron, you see that there are some periods with no action potentials in these neurons, and this is nicely related to upper peaks of this scalp recorded EEG. And again, another short periods of intense activity in these neurons, and a relation with the lower peak of scalp recorded EEG or cortical recorded EEG in these dogs. And another period of no action potential, of inhibition, of silence, and an upper peak and in the scalp or cortical recorded EEG, and so on. So what is the neurophysiological lesson of this picture? That the rhythmic activity of cortical pyramidal neurons, but the same is true for thalamocortical neurons, this rhythmic activity generates periods of windows of cortical activation in which the cortical neurons can talk each other, can communicate, can transmit information in these short periods. And then there is a pause, a silence, a period of inhibition in the cerebral cortex. And then another period, another window of neuronal activity and possibility to transmit information, to elaborate information, and then another period of silence and cortical inhibition. So you see that it's a functional organization based on oscillation of cortical activation with periods of activity, periods of inhibition, periods of activity at a given frequency, and that in that short period can occur the transmission of information in relation to contents of information processing, sensory, motor, cognitive, autonomic, and so on. So the oscillations of neuronal activity is an important part of our understanding of brain functions, not only for wakefulness, light sleep, deep sleep, REM sleep and dreaming, but also for the higher functions of memory, of attention, of reasoning, of decision making, and so on. So one should always evaluate not only the action potentials per se, the frequency of action potentials, the code of frequency of action potentials to understand the brain functions, but also the synchronization of the neural activity as reflected by brain rhythms within given brain circuits with windows of for functional connectivity, neural transmission, and elaboration of information in different nodes of brain circuits. And the frequency of these oscillations, the duration, the short duration of these active periods and inhibitory periods can frame and provides units of information processing in the brain circuits. And so researchers should try to understand this, the meaning and the contribution for the information processing or for inhibition of the brain circuits of frequencies at alpha, beta, and other bands of electrophysiological activity in the brain. Okay, so I say that for this first recording this year, first lesson this year is all. Thanks for your attention.